is Professor Pesce, in your opinion, when will the omic, I don't know which is what it means, be used in daily practice? Which kidney disease will benefit most? So, Dr. Pesce.
Thank you. So this is a question that often, actually, I would say always comes up when we give a talk, anybody give a talk on omics and system biology, because there's probably too much research in it at the moment, rather than a real diagnostic usefulness at this moment. The thing is that omics, we have different kind of omics, as you might remember from my lesson, my lecture. And when we talk about omics, we are referring to genetic, which Adele has was describing a very detailed approach to explore the genetic makeup of an individual. So sequencing, which kind of sequencing we use, and so on. So that's the genetic. Then we have the transcriptomics, so the gene expression, which can also be studied with the sequencing approach using RNA-seq to study which gene is expressed in a specific cell or in a specific tissue in a disease state, for example, versus a control. Then we have proteomics, which studies the proteins in a setting, for example, of a tissue or a specific cell, and we have metabolomics. These are all let's say, features of the flow of biological information, which, as you know, goes from DNA to RNA to protein to metabolites. When we study a disease with this approach, what we're trying to have and what we want to have in the future is, and daily practice, as you were asking, is to capture a feature which consider the DNA, so the variants that Adele is studying in rare diseases. Gene expression, because DNA is not enough to explain a process. It can be the basis, but there's something more to that, which can be a gene which is expressed and is highly expressed in some diseases written with regard to, for example, I don't know, in case of IgA nephropathy with gross hematuria in the same individual, and in the same individual when it doesn't have gross hematuria, the gene expression varies a lot, we found in a paper. So there's some gene which changes their expression. It's not related to the variation, a genetic problem, but more on a gene expression level. So third level, the protein levels. So for example, in the urine, we can find some specific protein which is altered in case of, for example, diabetic nephropathy. So, and so on. So each of these four compartments can have specific features, specific signature that change in a disease state. What we want to do in the future, in the daily practice, is like having a sort of barcode which captures the variability of each of these flow of information in the DNA, to study them all together. So for example, studying the DNA, the gene expression, for example, of some specific gene, the protein in the urine and the metabolites in the urine, putting them all together and have a very precise definition of the phenotype of an individual, which is the so-called stratification approach. So that's why, for example, a specific phenotype, it can be related to some specific combination of these four omics elements. So for example, when we have the ideal setting would be, let's say, diabetic nephropathy. No? Francesco is very expert, as far as I know, on this topic. So diabetic nephropathy, as you know, just to be, so that I can tell you something that you can relate what I'm talking about. As you know, can have real diabetic nephropathy, non-diabetic renal damage, which is not diabetic nephropathy, but some other kind of diabetic damage, and vascular damage, for example. So you can have different phenotypes of kidney damage on the biopsy for the same individual, but is still a diabetic patient, but the diabetic damage is very different. So you have to perform the biopsy to understand whether it's real diabetic nephropathy, non-diabetic renal damage, different damage from diabetic nephropathy, or only pure vascular damage. So to this purpose, in the future, instead of performing the renal biopsy, if we have a specific signature for in terms of genetics variation, so what Adele does, gene expression, for example, at the level of the peripheral blood mononuclear cell or in the urine, in terms of protein in the urine, in terms of metabolite in the urine, that captures and exactly classify the patient into these three categories. In the future, as you might imagine, you can ideally, in the very probably, like I would say next decade or so, at least reserve the kidney biopsies for those very strange cases that really needs to perform some kind of more invasive procedure to have a diagnosis. Otherwise, you can stop on using this signature, multi-omic signature, to identify this subcategory of diabetic patients. So that's the ideal setting that we want to move into which we want to move in the next future for daily clinical practice. Diabetic nephropathy is a good example for that.